Maybe the very first remark I would like to make, which is an obvious one, but I think from time to time it's good to repeat it. That is, if you look at where the advances came from, they come from basic science. We should not forget that. I mean, think about CRISPR-Cas9, think about siRNA, think about checkpoints of immune responses, and so on. They were all discoveries from basic scientists, and so this is important to remember. And for politicians, this is important to tell them that to support basic science is probably the best way to get at the end new drugs and advances in medicine. Since I know better gene therapy than any other field, I will focus most of my remarks on gene therapy, although I think some of them may be applicable to cell therapy or RNA therapy too. Maybe an interesting observation to make today, I mean, with the advances in the field, is that we are shifting a little bit from rare diseases to more common diseases, which I think is, of course, extremely significant for the future. But let's briefly examine what has been done in gene therapy. The most obvious important development is the one with CAR T cells, with the use of synthetic genes, of course, as you know, and this is a major advance over the last 10 years to treat a variety of B-cell malignancies. I don't need to go into detail, but as you know, there are 6 drugs today that have been commercialized, and I think worldwide more than 20,000 patients have been treated. In France, I think we are close to 1,000 a year, which is quite significant and raises a number of questions. I will come back to these questions at the end of my talk. And obviously, there are many perspectives in the field of CAR T-cell therapy, of course, to increase efficacy, to reduce toxicity, which is still an issue. Of course, the next challenge is to make them useful to treat some solid tumors. I think there are a little bit of advances there, but this is not yet achieved. And I think, and this is very exciting, and I think you talked this morning, if I'm correct, about the development of CAR T cells to treat autoimmune diseases, at least B-cell antibody-mediated autoimmune diseases. I think this is extremely exciting, and I'm pretty convinced that this will be a field of development in the next years to come. Then comes the treatment of genetic diseases. Today, in the last 25 years or so, approximately 20 inherited monogenic diseases have been treated with significant clinical benefit for patients, not always a cure, but at least a benefit. So in the field of ex vivo gene therapy with lentiviral vectors, this includes I think now 7 primary immunodeficiency diseases, so some metabolic diseases, some of them very difficult to treat, like metachromatic leukodystrophy or mucopolysaccharidosis, or then the advances are extremely dramatic. And of course, also the treatment of sickle cell disease and beta thalassemia, where classic gene addition by lentiviral vectors have had some effect. And of course, then comes the in vivo gene therapy with AAVs. As you know, significant results have been obtained in the treatment of hemophilias, of liver disease, of spinal muscular atrophy, and this is quite spectacular. But there are still quite a number of limitations, despite this significant number of successes. One is manufacturing. I mean, we hear just now some interesting talk about how to manufacture cells or vectors. And I think the issue of production of vectors, and for AAVs we need usually large quantities, remains a difficulty, as well as the vector is prepared for ex vivo gene transfer, the ex vivo transduction manipulation or manufacturing is still quite cumbersome, and if it can be simplified, it will be an advance. And we need, obviously, to go to automation. And I think the last talk was on this prospect significant. Another topic of interest where there are still limitations, if you think about ex vivo gene transfer and infusion into patients of stem cells or T lymphocytes, then you need the selection process, and this is usually done with chemotherapy, which can be done, but obviously which can be toxic, and probably there are some tricks, especially in the field of hematopoietic stem cells, may be used in the future to avoid to use any more chemotherapy, but rather, for instance, antibody or molecules that will attract the stem cells into the niches, into the bone marrow. With AAVs, there are some quite significant issues. I mentioned the need for, at least for neuromuscular diseases, to use quite a high dosage of vector, and this is a problem because of toxicity. I mean, this has been clearly shown in a recent past, where there are two problems, which is immediate toxicity caused by innate immune response that can be dramatic, and some patients died from it. And then secondarily, there is the immunogenicity of the vectors that play a role in limiting reutilization of such vectors. So these issues, and I think this has been a little bit underestimated over the past years, but today I think the questions of immune responses altogether, both innate and adaptive to AAVs, have to be tackled for future development. But of course, then comes the new steps, I would say, the second stage of gene therapy with genome editing, and I'm sure, of course, you discussed it, and based on the CRISPR-Cas9 system. And of course, the system per se, as you know, can be used to inactivate genes or regulatory elements of DNA, and this has been used with a spectacular success to the treatment of sickle cell disease and beta thalassemia, and there is now a drug approved that can be used to reinduce expression of fetal hemoglobin. This is an extremely elegant approach, which looks quite good. And some other examples come with the downregulation of kallikrein in patients with onconeurotic edema. And maybe this is a field where the more frequent diseases will be tackled, like hypercholesterolemia, because if you reduce the expression of PCSK9, this molecule that is involved in the metabolism of the bad cholesterol, you may control hypercholesterolemia, which is, of course, not a rare disease. But as you know very well, the CRISPR-Cas9 system as such cannot be used in other instances because it induces a number of targets. So, but there is now the second generation of genome editing that is coming, that has already been used in patients with several approaches. The base editing, which is the first one, with the modification of nucleotides based on usage of deaminases associated with NIC Cas9, so preventing the development of double-strand breaks into DNA, which are causing the problems related to CRISPR-Cas9. But beyond base editing, there is prime editing, where you can introduce into the DNA some stretch of a DNA sequence based on adding a reverse transcriptase into the guided RNA. And further developments may come for even more subtle or sophisticated strategy like paste editing with an integrase, or even transposon addition to introduce a full gene or quite a long sequence of DNA into the genome. And there are pioneers like David Liu at MIT that are developing these strategies that I think are extremely interesting and will lead to a number of clinical applications beyond base editing. But delivery is another important aspect, delivery of the genome editing products, but also the delivery, as I mentioned, of more classical DNA approaches. But if we can get rid of viral vectors, and I mentioned the problem with AAV, this would be fantastic. And this was mentioned earlier this afternoon, the usage of nanoparticles, lipid nanoparticles with ionosable lipids is of major interest, provided that, I mean, for some of the applications where there is a need to target the particles to the cell of interest, this could be achieved by adding at the surface of the lipid particles an antibody or a protein that will target the particle to the cell. I mean, there has been recently a publication about targeting hematopoietic stem cells in mice that looks promising, but we need to see further development. So there is a need to fund research. There are plenty of opportunities, a lot of questions to be solved, but again, with many potential applications. So it's great that this is being developed today in France with the, at least to support bioproduction, but not only bioproduction should be supported. I think research prior to go to the stage of bioproduction has to be quite significantly supported in order to be innovative and really to get rise, to develop new products. Okay, but then I'd like to finish with by developing a question which I think is an issue about the future development of cell therapy, gene therapy, RNA therapy, whatever, these new biotherapies, which is the price of these innovative treatments. I'm sure everyone is aware that all of these drugs, those that have been now on the market, I think there are 16 or 17 in the field of gene therapy, they have all a very, very high price. So the cheapest, if I may say so, are the CAR T cells with approximately 350,000 euros, but all others gene therapy are these days in the order of magnitude of 2 million euros, which is extremely expensive and by far the most expensive drugs that are on the market. So this is based on grounds that are, to my opinion, debatable. The three grounds are, one, these are sophisticated products. The second one is that usually the market is small, and the third one is that these are transformative therapies. But I would like to argue that, okay, this is more sophisticated usually than classical chemicals, but in most of the cases, if not all for gene therapy, the products have been conceived and tested up to the first clinical trials in academia or with the help of charities, so that the pharmas then, whatever they were, first startup and then bigger pharmas, they are reducing the risk by buying a product that has been quite advanced already. The size of the market does not seem to influence the price, so I don't think this is an acceptable argument. And the last one is transformative therapy. This is something of importance. Nevertheless, one should consider the fact that for most of these diseases treated up to now, whatever these are, cancer or genetic diseases, this is not always a cure. I mean, patients still require quite a number of treatments and care. So this is not all or nothing. And especially there are sequelae of neuromuscular diseases, for instance, and so on. And even if it would be the case, maybe the question should be raised, if there is a benefit, should the benefits go all to the pharma, or should it be shared in a reasonable way between the pharma and the society? I think this is an important question. So there are consequences of this. In the US, there is, as you know, very little regulation of prices, so that the prices are extremely high. In Europe, it is not so. This is not so, but still, the pressure comes from the US. But it leads to the fact that, for instance, one company, Bluebird, involved in the development of gene therapy products, has decided not to come to the European market because they consider that the prices that they will get is not high enough. Of course, this is a problem in terms of accessibility of these drugs for patients. So what can be done? Is it possible, is it feasible to reduce prices? Well, technically speaking, I would say there are some points to be discussed. First of all, with time, we may hope that the prices of a given drug will go down, although there are examples in both biotherapy where this is not the case, if I can develop if you wish. We can hope that automation and maybe regulatory processes that are slightly less constraining may lead to price reduction, let's hope, but this is not enough. And I think we have also to really consider the economical issue. And then I would like to briefly discuss, I am at the end of my time, the Barcelona case. I mean, probably you know that in one hospital in Barcelona, the people there, they have developed a facility to make their own CAR T cells that are delivered to patients, and the price at which the product is paid by the Social Security in Spain is 90,000 euros. So that is 3 to 4 times less than the prices from the CAR T cells developed by companies. So this is a priority medicine designation system that works in Spain. So it means, I mean, these simple examples shows that it is possible to pay less for a given sophisticated product. And we have to work on that. We have to work on that, I guess, by duplicating, developing such initiatives like the one in Barcelona. And I know there are some attempts to do it in other places in Europe. And I think we need to have not-for-profit production units of course, GMP grade drugs for gene therapy. This is absolutely required for the treatment of ultra-rare diseases, but perhaps also for less rare diseases. So I'm very much in favor of thinking about how to put together investments from the public as well as foundations and charities to develop such units. I think another aspect, and we're almost done, is that instead of having price negotiations at the national level, and I know that in France they do what they can within the CEPS, but if it will be done, for instance, at the European level, associated with EMA, I guess the strength in negotiation with pharmas will be stronger and cheaper prices can be achieved. I mean, there is an example of the feasibility of that approach. There are the vaccines against COVID. They were negotiated at the level of the European Union, and it works extremely well. So I think this is not easy, of course, to organize, politically speaking, but I think this is a way to go. And perhaps, by the way, more transparency, worldwide negotiation, that's, of course, very much more difficult with pharmas, might also lead to some reduction in prices. But in any case, I think it becomes urgent, given the fantastic opportunities that are developing in these different fields of biotherapies, to tackle this issue of prices if we want these medical innovations to profit everyone, at least in developed countries. Thank you for your attention.